Second session. Okay, thank you. Welcome to the second session of these online cell modeling seminars. Today, the topic is going to be modeling languages. Just to mention that this has been organized by the Center for Reproducible Biomedical Modeling, specifically by myself, but also Veronica and Joseph that are in the audience now. So anything that you will need, just tell us. So just to introduce the topic and the motivation of these seminars, probably you are already familiar with the workflow that we have to follow when we want to build a model. Starting with the biology knowledge that we have, we introduce everything in what we call models. And then the interest is to try to have engineering, design, and prediction functions in order to have applications. But of course, numerous challenges have to be addressed in order to have efficient models. And this is basically the motivation behind these seminars, that is to cover what we consider the 5 most important elements when we try to build a model. We already started with parameter estimation, but you have here the calendar for the rest of sessions, and we invite you to join us in 1 month and 2 months after, etc. So just for the people that are new here, or as a reminder, the structure of the seminar will start with an introduction of the challenge of 5 minutes. Then we will proceed with our first workshop, 20 minutes, the second workshop, and then a group discussion of 25 minutes. The goals that we would like to reminder for you is that we are interested in identifying common challenges in the field, share the personal experience and solutions that we have been working with, brainstorm new solutions, and more interesting for us, create a collaborative community of modelers. For that last part of the group discussion, is recommended to comment, always welcome. We want to promote the honest debate always, and remember please to mute your microphones when not talking. So starting with today's challenge, probably you are all familiar with this. It's just a basic enzymatic reaction. And probably you should be able to write it in this way, to represent it just without this system. This is a very cool way to represent and to model stuff. But of course, when you try to add more complexity to the system, for example, simulating a pathway, like in this case, the caspase activation, everything can be a little bit more complex. But imagine if you try to model more complex stuff like a whole cell, or even more complex like the human species. So basically, we took advantage of the software engineering field to take what they call modeling languages, that are artificial languages to express knowledge in a structured and defined way using consistent rules, in order to cover all the elements that are required when we are running biological models, from the topology, the networking, parameter definition, or even validation, to define what we call biological modeling languages that are specifically specialized in defining, networking, editing, and delivering these models. So it's not like there's only one solution. There are multiple variables for that. Probably you are familiar with some of these modeling languages that are specific for biology. There are some of them that are graphical, like the ones here, Mobi, Cell Designer, GenoCat. Others that are more text-based, like PySB, Kappa Language, BBSPICE, BioNetGen. But there are other differences that are interesting. Like, for example, there are specific ones that are non-executable, that they only serve to produce the model, like most of the ones here, but others that are executable and allow you to run and simulate the model. You can also take different model languages based on the basic language that they are coming from. For example, PySB comes from Python, BBSPICE comes from Spice, SynBiology is based on MATLAB. And the biggest difference at the level of application will be in the context for which they are defined. For example, we can have modeling languages more applied for the field of protein-protein interaction, and others more for the genetic circuit problems. So at the end, the goal is to take these concepts from engineering, software engineering, that are basically abstraction, reusability, object-oriented programming, inheritance, hierarchies, multiplicity, and extract interesting applications for the field of biology, like having a schematic and concise representation of the biology of our model, combine processes in a seamless manner, compatibility with all the stochastic simulations at the same time, share our models, etc. So to talk about this today, we have Professor Carlos Lopez from Department of Biochemistry at Vanderbilt University, who will talk about PySB, that is a framework based in Python to run rule-based modeling. And we also have Professor Morgan Madec from University of Strasbourg, who will talk about an extension of SPICE, which is a common language in electrical engineering fields, but extended to be used with biology models. So with this, I just welcome the first speaker, that I think will be Carlos, following the order presented here. So if you want to start to share your screen.
There you go. Thank you. Thank you so much. Thank you for the introduction. This is actually really cool. You already introduced half of my topic, so that's great. So I appreciate it. Let me share my screen then. Oh, you have to stop sharing your screen.
Yeah, I'm trying stop share. Okay, ready.
Let me try this. There we go. Okay, and then what I will do also is I will maybe turn off my video so that you guys can focus better on what I'm talking about. Okay, so thank you for inviting me for this. I was actually really excited to see this and to help out with this. And I think that there's a need for our community to be built around this kind of work. So thanks to the organizers for doing this for us, and Samuel for working all this out. So today I will talk to you about a little bit. I'll try to capture a little bit of what we're trying to do with PySB and how we're trying to model, to use Python as a programming language to kind of really program how cells work, cell behavior, cell dynamics, cell signaling. And I'll give you guys examples of what this looks like. Really our challenge or our goal is to model complex systems. And so that could be signaling processes, or cell-cell population dynamics, or anything where ODEs or stochastic differential equations or anything like that, anywhere where there's a dynamic process, PySB could, in theory, handle things like that. And as I was introducing earlier, the problem is relatively straightforward. We have a mass action kinetics, reactions like enzyme per substrate goes to some complex. The complex goes to an enzyme and a product. This is the classical Michaelis-Menten example. It has a forward and a reverse rate. And what people traditionally do is they write these equations like differential equations, where each component, each chemical species in this chemical reaction has a differential equation term. So dE time, ds time, and so on and so forth. And the parameters come in to describe the dynamics of the system. And you can do this very well and very easily. And then you end up with something that looks like this graph on the right, where you can make simulations of the system over time, where you can see how the substrate is consumed, or how the product is formed, or for example, the dynamics of the enzyme and the substrate, and how they all depend over time. But biology, as we all know, is a bit more complex than that. For example, here, this is an example from one of my postdocs, Leonard Harris, who has been working a lot with BioNetGen. And the idea here is that, for example, we have a complex. So biology happens a lot in complexes, right? This is an example where from this paper from Linov in 2006, where you have EGF bound to the receptor, and the receptor is bound to GRAB2 and SAS, and the receptor can also bind SHIC, and then they can also have different phosphorylation states. And so the field has worried or focused a lot on how do you represent these things. And so if you look at this in terms of how would you represent this in a graph, say, you would have something like the receptor, and the receptor can either be unbound or bound to EGF. The receptor can be either phosphorylated or unphosphorylated. If it's phosphorylated, it's bound to either nothing or GRAB2, and then GRAB2 can in itself be bound to nothing or bound to SAS. And the same thing for this other branch here, for this other binding site, where it can be phosphorylated or unphosphorylated, bound to SHIC, bound to phosphorylated, bound to GRB2, and so on. And so this gives a lot of potential combinations that leads to, for example, in this case, almost 1,200 states, which is a lot of states. And so when I started my postdoc, I was faced with something that looks exactly like this. So the postdoc that was with me, one of the guys that worked in this model from Albeck and Sorger, gave me this model, and he said, okay, good luck, see you later. And I was left with this thing that I had no idea what this meant. So K69 or K20, the biology is no longer here. And the question is, can we do this better? What is it about this that we can, how do we deal with this? And the main challenges for me were about, for example, model reuse. We cannot reuse this. We cannot share this. In a way, it became a dead end in the lab for how the models were used. And I said, well, there has to be some better way to do this. So this is where and I'll tell a bit about my background because I think this matters in terms of how we approach this. So I was trained as a physical chemist, and I did a lot of condensed matter physics, molecular simulation, molecular modeling, an area where there's a lot of automation developed around how models are developed and how models are implemented. And so at this time, I discovered, as Samuel mentioned earlier, BioNetGen and Kappa, which are languages that are so-called rules-based models, rules-based methods. And so again, to go to this example of Michaelis-Menten idea, our challenge was, well, can we take this set of equations, right, over here and turn them into something that the computer can read, and then use that to make building models and using models more like programming and less like having to write things by hand. So this is just an example of a rule for the same example for the enzyme for the Michaelis-Menten model. This is actual PySB language. This is actual Python. You define a rule, and you can and I'll tell you because it's a Python because Python is an object-oriented language. These are all objects. So the rule for this is, for example, the rule for the complex formation. There's an enzyme and a substrate that makes a complex described here by the % sign, and then the complex which is bound described here by these labels can become the enzyme and the product. And we can write this. We can look at this, explore this, and do something useful with this. Just to kind of walk you through this, this is what a rule looks like. And this is, I think, very common with BioNetGen and Kappa and other programming languages, other rules-based languages. There's a rule name. There has to be some name. There's a monomer name. You have to instantiate your monomers in some way. There's a site. They can be either state sites or binding sites. The sites have an attribute. In this case, non or bound to something. There's an operator. In this case, pipe has become the reversible reaction operator. This is for reasons associated with the Python programming language mostly. This is our binding symbol. The label indicates that they're bound or unbound. And then these are the parameters. And these parameters can be either single values or list of values. They can be as coarse-grained as you want or as well-defined as you want. And so with this, we can then go and expand this and start to functionalize this. And so that's what we did. We said, okay, now that we have these rules implemented in Python, and now that we have this system implemented in Python, what can we do with this? So this is where being in biology and having worked in a wet lab for almost 5 years really kind of changed my perspective on this. Because when I talk to the biologists, when I talk with my collaborators, they talk about actions. And so, for example, in this little schematic here, which is a schematic for apoptosis execution for mitochondrial permeabilization specifically, they talk about, for example, BAX translocates to the membrane, BAX oligomerizes and makes a pore, BID activates BAX, BCL2, MCL1, and BCLXL inhibit BID. And to me, all of these actions, translocate, oligomerize, inhibit, activate, they imply a function. And once they imply a function, to me, that's something that you could encode in a computer. So we basically took the rules and embedded in the Python programming language, and we basically supercharged them. So we embedded this, and we started to define functions like the translocate function, the oligomerize function, the inhibit, the activate function. And these functions take, for example, a target protein, an activator, a forward rate, a reverse rate, and then this dot dot represents the rules that can be functionalized. So now defining a model no longer is a matter of describing a bunch of equations, but rather function calls, right? And this becomes really useful when you deal with really large, complex molecular interactions, like for example, oligomerization. So in our case, BACs can become a monomer, can start as a monomer. Once it's activated, become a dimer, it can become a trimer, tetramers, and so on and so forth. Some people think it can even become up to a decamer. So rather than having to write these rules by hand, we just define a pore assembly rule, which says take a subunit, give me a size, and then just increase the size and write the rules for this. So we functionalize this. We make basically a loop, a Python loop, and we say this is our base unit, add a unit, there's a new pore, and then just append the rule, and then return the rules. And where this becomes really powerful is that this is our function call, pore assembly for BACs, make it a tetramer, and this list can be, again, as fine-grained or as coarse-grained as you need. If you want to have individual parameters for each reaction, you can do that. If you want to have one generic parameter for the whole system, you can do that as well. And with this one macro, generates 45 rules, which generates 150 ODs. And this is where you can start to really functionalize and encode programmatically the kind of a biology that you really need. And that's kind of what we're trying to do. So we like to think that these macros, functionalizing, treating these things as objects, as functional objects, allows the biology to be encoded efficiently, and this enables us to sort of build a sort of hierarchy, and we call it the recap of the approach, where at the bottom layer here we have the equations. These are the actual equations that you would write by hand. The rules are what PySB, Kappa, all those languages give us, which are the basic reactions or the patterns of those reactions. And then where PySB comes in is we take these objects and we make macros, which are in our parlance common biochemical processes, verbs in a word model, if you will. And then we from those macros we can make motifs, which would be, for example, small mechanistic elements or sentences in a word model. So, for example, bit activates the effectors, or sensitizers bind antipoptotics, the kind of things that you read in a paper that express actions. And then those things can in turn be embedded into modules. In this case, for example, in this paper in particular, we embedded this into modules that represented the apoptosis pathways, the direct, the indirect, the embedded pathway. And then we can start to build to build programs of what this biology really means. And I invite you to check out my lab website, where we, and our GitHub repository, where we now have a collection of about 40 or so different models that we can use. So to kind of place everything in context, so I think that, of course, as Samuel was saying at the beginning, no approach is as a panacea. No, we cannot do everything. There's many tools like MATLAB and Copacity that kind of deal with equations and reactions really well. This is just two of many, like you mentioned at the beginning. And then there's rules-based models, which kind of deal with reactions and rules. And where we sit around, it's really kind of this gray area where we're spanning a bunch of things. But the advantage of what we can do is really how do we encode complexity. So just to give you an idea of how this works, this is a depiction again from this paper about how PySB works under the hood. So here on the left, I have a model. Again, the ligand receptor binding. This is just a very simple model. This is my Python import and statement so that I can import the right things that I want. Then I declare my model. This can be a simple statement like this, or it could be files that you can reuse. And then once you declare once you make those declarations, the program will take these declarations, and we actually use BNG or Kappa to build the reaction network. Once we have built that reaction network, we take the graph generated by these tools, and we make the mathematical representation. In this case, it's ODEs. But as I will show you later, this could be Boolean logic rules, it could be stochastic differential equations, it could be fuzzy logic, it could be many different things. Once this mathematical representation is made, we can actually do something with this. In this case, we take advantage of the Python scientific computing environment, and then we use Bode for this example, and we execute the data, return the results, and then we can use, for example, matplotlib to plot the tools. What you're seeing here is really an open source and free version of MATLAB. This is a model that you can interact with, that you can play with, and you can use any of the tools available in the Python scientific community, which last time I checked was, there were, I think, under 100,000 or so Python packages in the Python repository. So we built this really cool, I think, environment for modeling, which goes beyond just a specification, but rather we want to make it into an environment, a way to really carry out biology, change the paradigm. So what are the benefits of this approach? So for one, we can, for example, use Jupyter Notebooks. So Jupyter Notebooks enable us to use literate modeling. This was introduced by what's his name, Knut, many years ago. And the idea was, could we make the code the document? So by using Jupyter Notebooks, we can make the code, run it in a web browser, run it locally. The results and the graphics are shown with the code. These notebooks can be version controlled. This helps with reproducibility, transparency. I don't have to just give you my method section in my paper, but I can give you my notebook, and you can actually check that and run the exact same environment that I did. And again, I refer you guys to the PySB website for this kinds of things. We can actually leverage decades of research in computer science. We can, for example, do model introspection. Once we define a model, we can actually start to do things like, can I search my model for patterns or rules or motifs that I want to evaluate or change? So in this case, for example, this is I'm saying, okay, in the module called AKT modules, extract all the catalysis patterns that contain the PTAN pattern, and it will give me all the complexes in the rules that have that function. So I can very easily probe and explore my model, and becomes a living process. One of the most useful things, and one of the things that we really try to emphasize, is this idea of, can we build upon models? So rather than having them be an end to the road, like I said initially, can we build on this? And to us, that implies having a way to check and make sure that our models are behaving the way that they should. So we've spent a lot of time building testing suites, and we've actually used the Python test suites and connected them to PySB. We could, for example, import we can build test suites where we could say, for example, okay, we're going to add a species to my system, and then we can check that the system is still working. And so it says, well, this species still exists. We've broken this binding site, so this failed, but the other tests passed, and we can actually check and make sure that the model still behaves the way that we want to. And we can do this with the model structure, with the model dynamics, with model outputs. Anything that you can convert into an object, you can check. And then the other thing that we want to do is really about, again, using the models. So one of the challenges that we found was that in many tools that have been very useful, like were mentioned initially, you're limited by what available simulation tools are, what simulation tools are available for your environment. So for example, if you're in MATLAB and you use the SimBiology workbench to build your tool, you're limited by whatever solvers are in MATLAB. And we wanted to break this mold. So what we did, we've spent a lot of time now, is building the simulation class, where we can instantiate a simulator object within the model, and then we can just we can set up all the parameters, and then we can choose a bunch of different conditions to run my model, and then use whatever the best mathematical representation I want for my simulation, and return the results. So right now, one of the advantages to doing this is that we're standardizing the interface so that you can use different simulation tools with the different models that you have. It can work with many simulation types, including stochastic and network-free. We take advantage of hardware, for example, multi-CPU, multi-GPU, hybrid environments, and we can easily run simulations across multiple parameter sets without boilerplate. We just supply an array. This has been incredibly powerful. So for example, this approach has helped us to, so here at Vanderbilt, for example, we have a cluster that's about, I think 10,000 or so processors, but it's shared by the university. And we had to do some simulations for a machine, that would require us to take the whole cluster for a month. And so they're not gonna let us use the whole cluster. So what we did instead is we actually took this and ran it on Amazon Web Services. So we made a container with the simulator class that we wanted. I think in this case it was Cup Soda. and we containerize this and split it across all the possible nodes on Amazon Web Services, and we used 5,000 processors for about a week, and we were able to get the results that we needed. So right now we can do ODEs, SSA, tau leaping, all of these tools from by NetGen. We can do GPU ODEs, GPU SSA. We're testing GPU tau leaping, and we're still developing this with a group in Italy, with a group of Marco Nobile and Daniela Bezzocchi. So and again, you know, the advantage of this kind of approach is that now things that were very difficult become trivial. So for example, things like model calibration, things like sensitivity analysis that take lots of computer resources. You know, running, for example, 16,000 simulations becomes a matter of, you know, less than an hour instead of taking, you know, months. And I refer you to some of our more recent work for this. Okay, then the other thing that we have been trying to deal with is, how do we share simulations? How do we share the model systems? How do we share simulation trajectories? These are hard to track. They take up a lot of disk space. We've leveraged the HDF5 based format where we can encode the data into hierarchical indexed compressed file formats that are binary, so we can store efficiently lots of data, and we can share efficiently lots of data. And in Python, this is very easy to manipulate, and you can load it into whatever database you're interested in. The last thing that I will touch on, and in the last 2 minutes, is visualization. One of the things that we found now is that, well, we want to be able to visualize our models and explore them and understand them, get a sort of feeling for what the models are doing. And so what we have done is we've made this tool, PyViper. This is currently under review, but you can check it out. There's a preprint in PyArchive. And the idea is that we can take this and do hierarchical exploration of models, and I'll let you kind of explore this. So in summary, PySB has access to a large software ecosystem for simulation, for visualization, for model calibration, for the model instantiation, and for analysis. And I won't touch on this in detail right now. There's not enough time, but please feel free to contact me if you want more information about this. And we're making a difference in terms of you know, real world examples. So we have, for example, PyDream, which is a model calibration tool, the model visualization, like I said, GPU-CPU hybrid simulation, and we've applied this to real systems like small allosteric simulations, small, large, complex systems. We can handle quite a bit. And then the last thing I want to end with is, you know, a lot of people say, well, that requires Python. So yes, you will have to learn Python, which can be a little bit of a challenge. It might take, you know, a week or so to kind of pick it up and get going. But in our opinion, it's really worth it, and what you will get out of this is really pretty significant. So I will leave it at that because I'm out of time. But you know, I'll be happy to answer questions at the end. Thank you.
Okay. Thank you very much, Carlos. Very nice presentation. So now I will introduce Professor Morgan Madec, who will talk about BBSPICE. Can you share the screen? And we start on questions, everything. We will leave it for the end with the group discussion. So if you have anything, you will have time later. Okay.
Is it okay?
Yeah, whenever you want.
Okay, thank you. So from my side, I will speak about software which is well known in microelectronics, which is SPICE. So it's a software used for the simulation of electronic circuit, and I will show you how to use this software to simulate biochemical and biological systems. Do you have on the screen? No.
Yeah,
okay.
It's working.
Yes. So just to start with the motivation of this work, I have a background in microelectronics, so I do my PhD 10 years ago in microelectronics, and we developed sensors, different kind of sensors. And in electronics, we like to model and to simulate the circuits before designing them. And we get some application in biological field, like for example, I don't know if you know this kind of sensor biomems, which consist of a microcantilever, and here you have some antibodies that catch some molecules, and when the molecules are catched by the antibodies, it bends the cantilever, and by piezoelectric effect you can measure the bending of this cantilever and measure the concentration of the target molecules. You have some application like living cell sensor, when you have here a transistor on which you grow some cells, and the cells change the properties of the transistor, and you can measure different biological signal by this way. And when you get interested on this application, the question was, okay, I have a complete environment to simulate my electronic circuit, the electronic part of my circuit. And I want to co-simulate the electronic and the biology that happened in my system, and I don't want to have two different softwares and to use two different software and environment to do that. I want to do that in a single, in a unique single environment. So that was our challenge, and that's the reason why we start working on BBSPICE. So first, just an introduction about SPICE language. So as I say in the first, SPICE language is well known in electronics and microelectronics. It's a software that is used for the simulation of electronic circuit, released in '71 and still used today for the design of electronic circuit. The idea is to describe an electronic circuit in a single text file, to give the user the ability to add new models, to describe new models, new circuit, to have a hierarchical description of the circuit. For example, you can use describe a part of the circuit and then consider this part of the circuit as a black box, and then you will have just one single model for the whole black box, and you can instantiate this black box in a top level description. There is a solver associated with SPICE that that first transform the description, the text file here in a set of differential equation according to the Kirchhoff's law. So the sum of the current that go into a node is equal to 0, and the sum of the voltage on a loop is equal to 0. This gives ordinary differential equations that are solved, and with SPICE you have a large range of analysis capability, transient simulation, static simulation, noise analysis, parameter estimation, parameter analysis, frequency analysis, etc., etc. And it's well adapted for the simulation of large-scale circuit, because now in electronics you may know that circuits have millions and billions of transistors inside. So SPICE is designed to simulate this circuit with many transistors. Regarding the description, the single file that is used for the description of the circuit, this file is quite easy because you have basically one line per device. So for example, here it's just a single circuit with one voltage source, one resistor, one capacitor. And I have just 3 lines. One to say that I have a voltage source connected between node 1 and node 0 and with some parameters. I have a resistor connected between node 1 and node 2, and I have a capacitor connected between node 2 and node 0. Okay, so that's, and the first word of each line is a label, and the first letter of the label corresponds to the type of device. So that's the basic on with SPICE. So now regarding the biology, so I will go fast on this part because it has been already explained. And everybody may know that. So you have the biological system that is described as an interaction graph like this, with the species, with the reaction, black arrow, and the regulation. And this leads to a ordinary differential equation. It's not the Michaelis-Menten equation, but it's also a classical system in synthetic biology. It's the toggle switch here. But you have this equation. And now what is very interesting is that you can draw an analogy between electronics and biology, because if you look at this equation here, the last one here, you can compare it to the equation of electronic circuits, assuming that the voltage is somehow equal to the concentration and that the current are somehow equivalent to the reaction rate. And this equation corresponds to a circuit with one capacitor, one resistor, and one current source. And this current source depends on another voltage, because the reaction rate depends on another concentration. So here the current source depends on another voltage. And in electronics, this device, a current source that depends and that can be controlled by another voltage, is a transistor. So we can take this equation and from this equation retrieve an electronic equivalent circuit composed of capacitor, resistor, and some transistors. So that's the first point. There is an analogy between this interaction graph and an electronic equivalent circuit. So we developed BBSPICE that translates this biological description into this electronic circuit without going through the ordinary differential equation. So now we need a way to describe the interaction graph. So that's the second point. So question is, what is the minimal set of information required to describe this system? And so that's something you may know too. You need the list of the species, and for each species, the name, the initial concentration, and the degradation rate. You need a list of the equation with for each equation an equation rate, the reaction, the product, etc., etc. You need some simulation directive, what you want to simulate, what are the conditions, and you need a list of parameters. So we formalize this description into a netlist, so again into a text file, which is very close to a SPICE netlist. So in this file, you will have first tag parameters, then the list of the parameters. Then you will have the species, here, a list of the species that are involved in this biological system, with for each the degradation rate and the initial concentration here. And then you will have the reaction. So this is the list of the reaction that corresponds to this graph. And we use the same trick as with as in SPICE. So each reaction, you have a label here. And the first letter of the label corresponds of the type of reaction. So for example, T means for transcription. That is to say that here we describe a reaction that is the transcription of the first gene. L means for translation, so that's the translation of mRNA into a protein. And B means for the binding. And then you have a specific here way to describe it. So for example, for the transcription, you need to give here the name of the mRNA that is transcribed, then a list of the activator, and then a list of the repressor. So in this case, I have no activator, but R2 played the role of a repressor for this reaction. And then here, the parameters. these parameters corresponds to the one you will find in the reaction rate. For the translation, it's just you give here the name of the translated molecule and the name of the mRNA that is used and the translation rate. And for the binding, you give here the reactant, here the product, and here the K-on and K-off parameters. So that's what I just explained here. And just to show you the equation, because we assume that the reaction rate of the transcription is always a Hill equation like this. So we just give here the name of the parameters that will fill this equation, but not the complete equation. There is a way to specify your own equation if you want. You start with the label X, and you give X the name of the reactant, the name of the product, the name of the modifier, and then you can write the reaction rate. So if it's not in the list, you have a list of 10 different kind of reaction. If it's not in the list, you can write your own equation. And finally, you give the simulation directive. Here, I want to simulate with a pulse of I1 here at this time, and then a pulse of I2 at this time. Then there is a Python script that will handle this description and translate it into a SPICE description. For example, here the parameters will be transformed into parameters in the SPICE syntax. For the reaction, for each species, you need to define one capacitor and one resistor. So for each species, you will find here a resistor, which value is the inverse of the degradation rate, and a capacitor, which value is 1, and the initial concentration, the initial condition correspond to the initial concentration. For the reaction, it's a little bit more tricky. You need to define the transistor in the electronic equivalent circuit. And here, for example, for one reaction, you will define 2 transistors because this reaction produces 2 mRNAs. So you will find here one transistor, the definition of one transistor that corresponds to the production of the first mRNA, and then another transistor for the production of the second mRNA. For the binding, here you will have 3 different transistors, one for the consumption of the first reactant, one for the consumption of the second reactant, and one for the production of the complex. And finally, the simulation directive will be transformed into SPICE simulation directives. And this is the complete environment. So you give the biological netlist in a text file, and finally you obtain a SIR file, which is a SPICE netlist that can be simulated with every SPICE distribution. I also developed some connection with SBML upstream and downstream. You can, instead of giving the model with our formalism, you can give the model as an SBML description. And from this description, you can also generate an SBML model, just to keep the compatibility with SBML. So everything is online on this address. I will show you here. So not this one, the other one here. So everything is online at this address. You can find the Python scripts. You can also find the examples, different examples, the benchmark that have been used for the validation, etc., etc. Just to show you how it works, I will try a live demo. I hope that everything will be okay. So the toggle switch, this is the description of the toggle switch in BBSPICE. So the parameters, the species, the reaction, the simulation. Then I open my Python environment, And here I just so this is just a script. You can compile a list of files. Here I just have the one single file. I run this this thing. So it recognized that in the BBSPICE description there are 6 parameters. There are 10 different species, 7 reaction, 2 DNA transcription, 3 mRNA translation, 2 binding, etc., etc. And then it generates the CR file with 6 parameters, 10 capacitors, 10 resistors, and 12 custom conductors, so 12 transistors. And it also generates an SBML file with 2 units, 6 parameters, 10 species, etc., etc. So this is the SBML model associated to this file, and this is the CR file with the parameters, capacitor, resistor, and the transistor here. And then I can use a SPICE distribution like this, ngSPICE, which is an open source SPICE distribution, and just write source and the name of the CR file, and it simulates my model. And here I have the results. So it corresponds to the results of the toggle switch, with in blue the output, in red the pulse of I1, and in green the pulse of the second molecule. So everything is available online. Regarding the reaction that are implemented, you can also read it on the article that has the paper that is associated with this work. And you have in this paper a list here of the different reaction that are implemented. The X, which corresponds to a custom reaction, in which you have to give the reaction rate, etc., etc., and a list of the simulation you can do. You can fix the concentration of a given value. You can generate pulse, waveforms, truth tables. You can simulate the steady state, the time course simulation. You can sweep some parameters, plot what you want, etc., etc. So I go back to my presentation. So that's the BBSPICE environment, and in the 2 last minutes, I will just show you some validation. So that's just what I show you in the live demo. What is interesting is the comparison of the computation time between SPICE and COPACY. So I simulate the same circuit, the same biological system with COPACY and SPICE. And you can see that with the increasing number of reactions, you have linear behavior of the computation time with ngSPICE, and it's not the case with COPACY. So if you have in your system 10,000 of reaction, there is more than 1 order of magnitude in terms of computation time between ngSPICE and COPACY. And for the steady state simulation, it's more spectacular, because even with low number of reaction, you already have 2 order of magnitude in terms of computation time. This is because the algorithm used by SPICE is very, very optimized, and is very optimized for large system and for steady state computation. It is more spectacular if you use, so this was the result with ngSPICE. Again, ngSPICE is an open source free SPICE simulator. If you use commercial SPICE simulators, like for example Spectre, which is used in the industry of microelectronics for the design of large scale circuit with many transistors, you can see that it's very, very spectacular. Here you have 1, 2, 3 order of magnitude in terms of computation time. So with SPICE, you can really simulate very large circuits with a very short computation time. Here, it's an artificial gene regulatory network with 10,000 of genes, and 10,000 of regulation, and you can reach obtain the result in 1 second with SPICE, whereas it takes 1 hour with Copacy. To finish, just I will show you what is the next step. The next step is to integrate 2D and 3-dimensional reaction diffusion model in also in SPICE, because in some system you need to introduce partial differential equation, and this has been done using an adaptation of an existing tool from microelectronics too, and the similitude between the Fick law, the modelization of molecular diffusion in biology, and the thermal diffusion in electronics. So basically, what we simulated is a system like this with 2 cells, one cells that synthesize an activator that flow to the other cells, and then it's activate a gene that synthesizes a repressor for the first cell. So it's an oscillatory behavior, and we mesh the space like this. This is the lattice used for the simulation, for the discretization of partial differential equation, and with SPICE we also obtain simulation result and can simulate the things like this. So to conclude, BBSPICE is a way to model and simulate biological system with SPICE. We check that we have comparable results in terms of accuracy in comparison with Copacy, for example, but with a speed up of 1, 2, or 3 order of magnitude if you use a very optimized SPICE distribution. So it's very interesting for us for the virtual prototyping of bioelectric electronic systems like biosensor. It could be a way to adapt electronic design automation tools to biology. It's an alternative to existing biological simulator for large-scale problems. We can implement 2D or 3-dimensional model. It's easy to manipulate by a third-party software and algorithm. That's very important, especially if you try to optimize your model, your system. You need to run many iterations, many times the model, changing just some parameters. So if you want to run the model with other parameters, you just have to change the text file. It's easy, and it allows multi-physics simulation, because with SPICE we can simulate electronics, we can simulate now biology, but we can also simulate thermal effect, we can simulate mechanics, we can simulate microfluidics, etc., etc. So thank you for your attention. I was a little bit long, but I hope that everything was clear. And again, will be happy to answer your questions.
Okay, thank you very much. So nice presentation. We will move now for. So if I can share my screen. Well, you have time now. Okay, there it is. We have time for discuss a little bit about this topic now. First, I will ask to the audience if there's any question for our invited speakers. Okay, so I will guide this discussion, starting first with specific questions for the model languages that have been presented. I think for those working in biology, it's more or less clear that there are more specific developed ones like PySB that seems to be more familiar for biologists, and others like BBSPICE that comes from an adapted, already existing language from engineering. So I would like to know if you have something to say about the advantages and disadvantages of each option in each case. So. Carlos, Morgan, can you hear me? So if you want to add something, if you feel like there's nothing to say about this, we can move. There are more questions.
Morgan, I'll let you take the lead.
I don't know if Morgan is here. Okay, let's see.
I think he's muted.
Yes.
Okay, now we can hear you.
Okay. I don't know if we can compare the two approaches, because it's as far as I understand, PSB is a way to generate. It's a way to describe and generate models. So, to my opinion, they are not comparable. They are complementary, because with the PSB you can generate easily very, very complex models, which is not the case with BBSPICE. But BBSPICE, the language is let's say straightforward enough to also write some scripts to generate automatically BBSPICE description.
I would add to that in my experience, most of these tools are usually more complementary than really competing. And I'm not just talking about BBSPICE, I'm talking about the ecosystem in general. I think that the motivation to develop different tools arises from a need, and they all meet different needs. I think that I think that Morgan mentioned this also. The commonality, I think, is, can we have a common interface to work with these things? I think that Python is slowly becoming that. And I think that, for example, when I was watching Morgan's presentation, to me, the immediate idea was, well, could I make very large models and use Morgan's Python interface to connect to BBSPICE and actually do something with Spice in a straightforward manner? And I think that's where the strength really lies in this approaches for modeling languages.
Cool. So probably the second question for this part has been already addressed during the presentation about the best biological cases. So related to what both of you mentioned is that probably one of the problems when you start to think about modeling something is which is going to be the best modeling language for your specific case. And there are times that it's very complicated to know if it's going to be adequate beforehand, because there are times that you just start to write things, and then when you have wasted already 1 week, you notice that it was for nothing because the most important step cannot be accomplished. So any recommendations about how to deal with these cases, if there's any like best practices when we choose the modeling language?
So I have a lot of opinions about this. So apologies to everyone, because I've suffered through a lot of this. I think that I think the main challenge is really what is the model, and then what is the modeling language that we're going to use. So I think the first question is, what do I want to model? Do I want to model activation, subpopulation dynamics? And then the language is, well, how do I want to encode that? How do I want to define those interactions? And I think different languages are better for different things. So if you think about, for example, programming languages, sometimes C or C++ or Fortran are very suitable for certain applications. And that's a useful language to describe, mathematics or manage hardware. But Python is a high-level language that helps you abstract concepts into executable chunks or executable objects. So I think that I think that getting at the point where we're even and Herbert just actually brought this up, getting to this point where we're saying, what is even a modeling language and how do we define that? I think that that's a much harder problem for biology. I think that we're borrowing different tools and different ideas from different fields, and we're slowly converging to that. So at least that's kind of my view. And I think that these things are, so it's more about, for example, in the case of PySB, if you're interested in dynamics of complex networks, PySB is a great tool. But if you want to do, I don't know, something very different that's more related to circuits, then something else would be better. I'm sure other people might want to comment to that.
Yes, and BBSPICE has an advantage is that it exploits the SPICE simulator, which exists from 50 years now, and which has been optimized for large-scale circuits with many nodes. And as you have seen in the presentation, you can have an improved simulation time in comparison with standard simulators. And that's the advantage. The disadvantage of BBSPICE is that the simulator is a black box. So I understand that with PySB you can tune the simulator, which requires some knowledge on mathematics and etc. But you can tune the simulator. You can adapt the simulator on what you want to simulate. With BBSPICE, you just have a black box. You describe your circuit. You don't know how it is modeled or how it is simulated. You just have to trust on the simulator and trust on the fact that it works. And that's it. And the second point is that with BBSPICE, we only have the deterministic simulation capability. So we cannot use stochastic simulation, for example, because in electronics we never use stochastic simulation. So that's one of the shortcomings of BBSPICE is that you have a simulator that simulates deterministically the equation, and it's a black box. You cannot change anything.
Okay, so I would like to move to the last block of questions that will be more related with the perspectives in the field. So I think having two experts will be interesting to ask the question if there's something impossible to represent by available modeling languages, not only BBSPICE or PySB, or what do you think would be a big challenge to represent using modeling languages?
Go ahead.
Yes. With up to now, I never encounter a system with equation that cannot be used in BBSPICE because there is some restriction about what are in the differential equation. For example, you can not have a derivative term on the right hand of the differential equation, because if you have this kind of with BBSPICE, if you have this kind of equation, you cannot find an electronic equivalent circuit, so you cannot use BBSPICE. But up to now, I never had this kind of, I don't know if there is some cases in biology in which you have this kind of equation, but up to now I did not find them. I look on the biomodel database. I don't know if you know this, but it's a database of large database of SBML model, and I tried to translate all the SBML model of this database into BBSPICE. I don't simulate all of them, but just try to translate them, and up to now I did not find any trouble. The other question is the modeling language suitable with a problem that depends on the location, on the space. Because most of the time you consider that in your system the concentration is homogeneous, and then you have ordinary differential equation, but sometimes you need to define the system with a partial differential equation, which is a challenge. With BBSPICE, we develop an extension, as I just mentioned in the last slide of my presentation, to handle this this thing. But but it's kind of, it's not very, it just use a finite differences method to solve the partial differential equation. It's not well optimized. So for me, that's the two limitation we can imagine for BBSPICE.
Okay. Same question?
Up to you, Carlos. If you like to add something to the answer,
sure.
You're welcome.
I think that the challenge will be in, as some people in the chat are mentioning, the challenge is also about, modeling is really about what models are you using. So I think if we're going to move towards modeling whole cells, cell populations, and then we get into the problems of scale, right? So do we need molecular or sub-molecular atomic representations to actually capture the intricacies of a given model system? That's where I think we will face a very significant challenge. Anything that involves crossing multiple scales, I think is going to be very hard. So for example, in my lab, we're thinking about, well, we get a lot of single cell data right now. And that means that a mechanistic model is better viewed as a probabilistic rather than a deterministic process. And then how do we impose that modeling representation onto a cell population? And that's where I think that things get a little bit more complicated. So I think scales is going to be the next challenge. And in reference to SBML, since it was both raised in the chat and your questions here, I think SBML is a great kind of start to standardize how we communicate. But I think there might be also limitations. So for example, one of the things we have found that can be very challenging is that SBML was defined to be, at least the first generations were a flat representation of the model system. But then how do you encode the hierarchy of complexity that comes from a biological system? This is how people think about biological systems. They think about the apoptosis signaling pathway, and then the apoptosis signaling pathway has sub-branches and sub-components. And that I think is going to be very hard to incorporate into one standard, and I don't know what that looks like yet. In computer science, they have chosen to just leave it to the code and document code very well. So I don't know what that means for us.
Okay, so I think you agree in that the problem of multi-scale. Do you know any language well suited for this? I'm asking the two guest speakers, but also audience, if there's anyone that wants to add something. Just mention it.
Just to plant a seed there. I worked on a project with Department of Defense, and I cannot tell too much details about it, but it was about multiple scales. And the idea was, well, if you have a living system, how do you describe that? So if you think about, for example, an airplane model, the engine is modeled one way, the wings are modeled another way, the ventilation system is modeled another way. How do you bring those together? And the development was precisely to develop platforms to bring multiple models together on a common system. I think we will need that at some point. So I think that as we get into more and more complex systems, we will need something along those lines. And no one has mentioned anything about model calibration, model parameter estimation. But I think that another big challenge is that we don't have enough data. Even with the tons of data that people collect right now, we still don't have enough data to correctly represent a biological system. So what is that going to mean looking forward? I think that's a field that we need to address.
Do you want to add any comment, Morgan?
Before we move too far forward, I noticed that Paul had his hand raised for a bit, so just wondering if Paul wanted to add a comment.
Yeah, sure. So just 5 minutes ago or so, we were talking about simulations, and I think it was last month when Victor Zavala gave his presentation about discretizing systems of ODEs, which then the solution will not longer require simulations, but it would be solved algebraically. So I was wondering if there's still a need for faster simulators, or if what Victor has proposed, if I understood him correctly, will always be, a faster solution.
I'm not familiar with that, so I'll have to look at it more. In principle, if we could solve these things algebraically, that would solve a lot of headaches. But at least based on what I know, I don't think we're going to get rid of solvers very soon. I don't know if anyone else in the audience might know more about this. Maybe you, Mark or Morgan.
From what I remember from Dr. Zavala's presentation, the actual algebraic solver was looking at just like ways that they can reduce the model, I guess, to see which parameters vary in accordance with other ones. But I think it wasn't actually able to like fully capture nonlinear behavior on its own. So you'd still have to run the simulator. I think it was just able to reduce the number of parameters that needed to be calibrated. Anyone can jump in and correct me if they have a different number.
Okay, so it's already time for finishing the meeting. Just to thank both the speakers again, Morgan Madec and Carlos Lopez. I'm sure that they will be happy if you contact them to answer any question that can be still there. So just to put again the first slide where I talk about the next meeting. So invite you in August 7th to the model validation topic with Rick Gerkin and Christian Leven. I hope to see all of you there. So thank you very much for coming to this meeting and see you in a month.
Thank you very much.
See you.
Thank you, guys.